Hello, this video is gonna give you an overview of Unit 1 and Unit 2 together. So let us look at what is fermentation. Fermentation is the process of using microorganisms to produce anything that is useful or economically important. So fermentation can be the process that is used to make idli, dosha, bread, to the process. Okay, we can use the term to define the process to produce recombinant vaccines, insulin, etc. So where all can fermentation be used? So it can be used to produce recombinant products. Recombinant products are here you'll be producing products, you'll be introducing a recombinant vector. You'll be having a vector, you'll be introducing a gene of interest. For example, into a vector, you can introduce a gene for insulin. Then this recombinant vector can be introduced into the bacteria. It is transformed into the bacteria is allowed to grow and insulin is produced. So this is recombinant products. Some examples are insulin, recombinant vaccines, growth hormone, interleukins, etc. You can use this procedure fermentation for producing microbial biomass. Here the final product is the microorganism or the microorganism biomass itself. Example, yeast production. So baker's brewer's yeast or nutritional yeast can be produced by fermentation technology. Then you can use it for biotransformation. So transformation means you're changing one product to another, you're transforming it. So this can be used to transform a chemical to a identical chemical. You can transform wine, that is alcohol, into vinegar, acetic acid, alcohol to acid transformation. Then you can use this process for producing metabolites, and metabolites can be primary metabolites which are produced by the microorganism, which is needed for its growth and development. When the microorganism is growing and dividing, it will need nucleic acid, it will need proteins for its cell membrane synthesis, it will need fatty acids for different purposes. Okay, it will need amino acid. So all this that is needed by the microorganism during its cell division and growth comes under primary metabolite. Then you can also use it to produce secondary metabolite. So secondary metabolites are those compounds which are produced by the microorganism and are not needed directly by the microorganism for its growth and development or growth and reproduction. So secondary metabolites are additional compound produced by the microorganism, like antibiotic. It is helping the microorganism to kill the invading or other microorganism. So secondary metabolites are usually produced in the late log phase or in the stationary phase, whereas primary metabolites are produced in the log phase when the cell is actively dividing. You can also use it to produce enzymes like amylase, protease, etc. So when you're looking at the whole process, it can be divided into upstream process where you are producing the media, you're formulating the media, you're growing the microorganism, you're selecting the microorganism that should be used for the process, you're like improving the microorganism, everything that is happening before your original process. Okay, then you'll have the process itself, that is whatever is happening within the bioreactor. The cell is growing, it is producing enzymes, the product is being formed. Sometimes you will need enzyme immobilization for certain bioreaction. All those steps that is happening during the process formation, product formation. And finally, you have the downstream process. That is after your bioreactor process is completed, you will have your resultant media with your product, with your cells, with the metabolites, extra additional cell debris, solid particles which were not used as spent substrate. So after you get this, you'll have to separate whatever you are needing. You'll have to extract it, you'll have to clean it, you'll have to concentrate and formulate it. That process happening after the bioreaction is called as downstream process. So first let's look at the upstream process. When you're looking at the upstream process, we are mostly interested on media preparation and microorganism that is used for fermentation. So media or raw material, for every microorganism to grow, it have to have some media. Okay, and in a bioreactor also you'll have to supplement it with the media. So the media can be crude, that is it is undefined, you don't know what is the actual constituent inside the media, or it can be synthetic defined media. You know, yes, this media is having so and so carbon, so and so nitrogen, it is like it's completely known. But when you're looking at the fermentation level, mostly crude medias are preferred. Why? Because it is economically more, it is having less cost, and the crude media like sugarcane molasses, then waste from different factories like sugar production, waste from sugar production, waste from milling of rice and wheat, waste from the milk industry like whey protein, all this can be considered as a crude media. So it is a rich source of carbon, nitrogen, and many inorganic and organic constituent. Then you can have the synthetic media also, but synthetic media are always preferred at the laboratory level. So what should be the content inside the media? It should have carbon source, nitrogen source, water, it should have buffering system, it should have antifoams, or if a microorganism is needing something extra, it should have all the things that is supporting the microorganism to grow, plus it is supporting whatever is to be produced. Then before you go for your process, the media should be sterilized. It should be made clean. Okay, only then you can prevent contamination. So the most preferred method for media sterilization is filter sterilization or heat sterilization. You can use different types of filters. Or heat sterilization can be done as a continuous heat sterilization or as a batches. So here you will be having heat exchangers to increase the heat and to sterilize the media. And usually steam is filtered steam or clean steam is the source of heat sterilization, or it is helping in heat sterilization. And when you're selecting the media, it will depend on two important factors. What is the type of product you're producing? If you're producing a primary metabolites, you want the microorganism to grow more because the primary metabolites are produced in the log phase. So you'll be giving the media such that it is promoting the microorganism's growth and division. If you're looking at the secondary metabolite, you will be supplementing the media such that the secondary metabolite is produced. For example, you will be providing some additional chemical that can be used as a raw material, and more amount of the secondary metabolite can be produced. The selection of media also depend on the type of organism you're using. Like if you're using an autotrophic microorganism which can produce its own food, then the media will be minimal media, just some inorganic components, water, carbon dioxide, etc. But you are going for organism which require extra supplement, which need something extra, then you'll have to supplement the media with all the requirement that the microorganism is having. Anything that the microorganism calls for, like additional amino acids, additional vitamins, or carbon, lactose, whatever source. Then if you're using a solid media, the fermentation you will be going for will be solid state fermentation, and here the media will be solid and the moisture content will be less than 15%. Usually solid state fermentation is done as batch fermentation, or else if you are having a liquid media, you will be going for submerged fermentation. The fermentation procedure will be submerged fermentation. Okay, and submerged fermentation can be done as continuous fermentation, batch fermentation, or fed batch fermentation, whereas solid state fermentation is only carried out as batch fermentation. Now you'll be looking at the second thing in the upstream process, that is a microorganism that is used for fermentation. We can use already reported microorganism for fermentation. They can be used, or else we can isolate more promising microorganism with a desired quality from the environment. And if you are going for the isolation of microorganism, you'll have to screen for microorganism from the environment, and the screening will be primary screening, where you're removing all the unwanted microorganism. You'll be having a group of microorganism, and you will be selecting only microorganism that is having the property you're looking for. All the others will be eliminated. And in the secondary screening, you will be looking at these microorganisms and knowing that which of these, even though it is having the property, it need not be industrially applicable. So you are selecting the industrially applicable microorganism. And you'll be looking at other factors rather than the productivity alone. You'll be looking at, is the microorganism growing easily? What is the media requirement for the microorganism? How well is it growing? Is it having, it is stable, is it stable or not? All this will also be checked in. Okay, and after primary and secondary screening, you can select the microorganism. Sometimes you can improve the strain also. After your screening, you're selecting a microorganism, you can introduce some sort of an improvement to this microorganism, or else the already reported microorganism. This can also be improved such that the production or productivity of the microorganism is enhanced. The microorganism is more fit for the bioreaction process. So strain improvement, how all can you do the strain improvement? Either you can go for induced mutation, you can induce a mutation, but mutation is random, so you don't know what is a mutation, like what is the change that is brought about by mutation. So after you induce your mutation, you will have to go for mutant selection. You'll have to select the mutant, you'll have to screen it, is it producing so and so, and to how much extent. Or else mutation is one method, the other method is recombinant DNA technology, by which you are introducing a new gene, or else you can go for recombination. Okay, and then any microorganism, okay, have to be preserved for further use such that it is not contaminated. It is having the original property, the chemical property, the biological property is maintained, and what, it is viable. So you'll have to preserve it in such a way. There are different preservation methods. Okay, and methods can be divided into preservation in its metabolically active form or metabolically suspended form. So different examples are, you can store it at low temperature, repeated subculture is a technique, overlaying with mineral oil, you can store it, it has a spore in the sterile soil, you can go for lyophilization, cryopreservation, etc. But anything you will have to go for quality check in between to make sure that the microorganism is viable, it is not contaminated, it is showing all the property, there is no mutation, and it is having the original properties. The next is bioreactor. So a bioreactor should have all the setup to help the fermentation to proceed normally. So these are the parts of the bioreactor. It should have a vessel for fermentation. Okay, it should be made with metal or glass. It should be able to withstand the microorganism growth. It should not be able to, it should not corrode easily. It should be able to withstand pressure, temperature, pH, etc. Then you have the aeration and agitation system. Aeration and agitation system includes the impellers, the baffles, and the sparger. Sparger is for providing air, impeller is for mixing, and the baffle is to uh, remove the vortex created by the mixing. Then you have feed port, whereby you feed the media, the microorganism, and you have smaller ports, that is inoculation port for microorganism, inlet for air. Then you have the acid and base inlet, whereby you will use it to maintain the pH, etc. Then you have the heating and cooling system. Heating coils will be there. Then it will have a what? It will have a cold water jacket to cool the system so that the temperature is maintained. You should have a foam control. So all together, all these are needed for a good bioreactor. And as the bioreaction is proceeding, the oxygen content, the pH, the temperature will start to change. But always for a microorganism, this should be maintained in a certain range so that the process is proceeding well and good. So there should be sensors to sense these variables, and there should be control system to control these. So you should be looking at the sensors and the control system. For example, for temperature, the sensors will be different types of thermometer, which is sensing the temperature, and then the control system would be the cooling and the heating coil and the cooling jacket. Then fermentation, when you're talking about fermentation, you can do fermentation. The process can be batch, it can be fed batch or continuous fermentation. And continuous fermentation, to maintain the fermentation in a continuous mode, you can use chemostats and turbidostat, which will regulate the addition and removal of media and the spent media respectively. You have different types of bioreactors, okay, depending on what is the type of mixing. What are you using? Impellers? Are you using air bubbles? Okay, is it what is the sparger system? Are you using like the immobilized enzymes, etc., the shape of the bioreactor. According to all this, you have different types of bioreactor: the continuous stirred bioreactor, the bubble column bioreactor, airlift bioreactor, fluidized bed bioreactor, packed bed bioreactor, and photobioreactor. After your process is complete, you'll have to go to the downstream processing. Here, the various process that is involved, this will have all the process that is involved in the recovery of your product. So after your fermentation is completed, you will have a media with your product, which will be at a very low concentration. The media will also have the cells, the used and unused media, some solid substances, raw materials, certain other byproducts, certain other metabolites, etc. So finally, you just want your product of interest. So you'll have to separate all this. So the steps are, first you will have to separate the liquid and the solid, that is the media, the solid part of the media, the cell have to be separated from the liquid. And if you are having intracellular product, that is the microorganism can either produce product inside itself, microorganism product, or else it can produce product extracellularly. After the product is produced, it is coming out of the cell wall or the cell membrane and it is released into the media. So if it is extracellular product, the product is inside the media. You don't have to go inside the microorganism. The extracellular product, you just have to go for solid liquid separation, concentration, purification, and formulation. But if it is intracellular product, you will also have to release the intracellular product. So looking at the steps in detail, first, looking at the intracellular product. The product is inside the cell, so the first step will be cell disruption. And cell can be broken up by different methods like mechanical, which is including physical methods. Okay, then you can go for chemical methods like SDS, CTAP, or other chemical that is breaking the cell wall. Then you can use enzymatic method, lysozyme, proteinases, etc. Mechanical and physical method like grinding, high pressure. All this can be used to break open the cell. After the cell is broken open, this whatever content was inside the cell, now it is released outside. So your product is also coming outside. Now you will have to remove the insoluble, or you will have to go for the solid liquid separation. So how do you remove the solid from the liquid? You can go for techniques like centrifugation, flocculation, flotation, filtration, etc., where you're separating your media containing your product from the solid particle. But if you are going for the biomass, if you are extracting the biomass, the solid separated will be your place of interest. Okay, because biomass means it is the cell. Then after you separate the solid and the liquid, now you're having a lot of liquid media in which you are having your product of interest. But the product of interest will be in a very, very low concentration. In 1,000 liters of the media, maybe 1 or 2 liter will be your product. So now the next step should be concentration of the media or isolation. Okay, so concentration of the media can be done by evaporation, by liquid extraction, by precipitation, by different types of filters. Now, in this step, you are removing the water content and you're reducing the water content and you're concentrating your product. The next step is purification. So, in purification, now you have a concentrated product, but you might have some impurities which are very, very similar to your product. That should also be removed so that you get a pure, highly pure product. And for this, the mostly used method is chromatography. So purification can be done by chromatography, but chromatography is a specialized technique. It is costly. So this purification method is always preferred for certain type of downstream processes. Not all the process, not all the product go through the all the 5 steps. Some might only require 1 or 2 of the steps, some might require all the steps, some might require combination of steps. Okay, so purification can be done by chromatography. And chromatography, there are different types of chromatography, like ion exchange chromatography, affinity chromatography, size exclusion chromatography, whereby you are separating your product from the mix using certain properties. And after you isolate or you purify your product, isolate and purify your product, now you will have to dry it, crystallize it, and formulate it. So you can go for drying by lyophilization, uh, uh, or spray dyeing. You can go for crystallization. Some product will have to be sterilized. All the step is done finally, and after all the step is done, your product is ready to go. So these are the complete processes. So each process you'll have to be studying, but this is a complete overview of chapter 1 and unit 1 and unit 2.